Hello, welcome to Medical Immunology. Today we will be discussing chimeric antigen receptor T cell immunotherapy for B cell acute lymphoblastic leukemia. The goal of today's lecture is to prepare you for the upcoming quiz. The quiz will be based off of Brentjens et al. The reading, this is going to be a very dense hematology oncology cellular-based therapy reading. Should be pretty interesting is my hope. For rules for this quiz, please access the Canvas page to look at instructions for this quiz, but this will be totally open book. In addition, this will be totally open to your classmates as well. What I mean by that is that this can be a group quiz, so you can work with your classmates and discuss the questions together. Once you start the quiz, you'll have an hour to finish. So please work together on this quiz, open book. The hope is that this quiz will be interesting, this paper will be interesting, and hopefully you can learn something as well. Specifically, we're interested in learning about drug development, and we're also interested in learning about hematology oncology, and we're also interested in learning about the clinical application of those basic science signaling pathways that we've been discussing over the past couple lectures. So this is the drug development regulatory path. So anytime there's a new therapeutic that enters the market, a new drug that can be prescribed at the pharmacy counter, is that all of those drugs have gone through this quite complex stage of clinical trials and preclinical research. So starting with the preclinical research, this involves identifying novel drug targets. So these are typically proteins or nucleotides or something within the body that can be drugged. So identifying something that's important for the induction of disease or something that has gone wrong in certain disease states and seeing, can I develop something that can alter this? Can I develop something that can change this? And the way we do that is we use basic research. So basic research helped us identify molecules like CD19 that are targeted here. It also helped us identify these signaling pathways that are changed in these immune cells in different immune settings. This involves a lot of chemistry and biology. In this paper, there was a lot of molecular biology, developing these chimeric antigen receptors, developing vectors where I can take these things and put them into different T cells to change the composition of those T cells to make those go out and fight cancer. Ultimately, there's animal testing. Animal testing is going to be going and taking these and putting them into rodents, putting them into larger animals like dogs or pigs or monkeys and seeing, do these work to kill cancer in those animals? Ultimately, once we have a critical mass of data, both for safety and efficacy and pharmacology and manufacturing, from there we can go to the regulatory body, the US Food and Drug Administration, and we can ask for an IND, an investigational new drug application. The investigational new drug application will give us permission to go into first-in-human testing. This is going to be your phase 1 clinical trial. Today's study is a phase 1 clinical trial looking at this T cell therapy. Even though this isn't a first-in-human trial, this is going to be the first time that this drug was used on this particular patient population. For phase 1 trials, the primary clinical endpoint is going to be your safety testing. Typically, this is done in healthy controls. When I was in grad school, I would sign up as a volunteer, or not a volunteer, but I would sign up to take novel experimental therapeutics as part of phase 1 clinical trials, and that's how I would get additional compensation. There's a number of clinical trial sites in Orange County, in LA County, that bring people in, and you can sign people up to do these healthy control and try new experimental medicines. And I did that for a number of years until I had an adverse event with one of those medicines, and I decided it probably wasn't worth my time anymore. In the case of this clinical trial, we can look at refractory patient efficacy. In this case, these are oncology patients who are entirely refractory to standards of care. So these are chemotherapy refractory patients that have B-cell acute lymphoblastic leukemias. So with this, this is kind of the framework for what we're going to be studying today. After you get good efficacy and safety in a phase 1 clinical setting, from there you can advance to a phase 2 clinical trial, which has more patients. This is primarily going to be focused on measuring the clinical efficacy in a large patient population. You're also going to continue to monitor safety. In addition, in a phase 3 clinical trial, we're going to look at efficacy in a large patient population. We're also going to try to look for more rare adverse events and also compare this to standards of care. So typically trying to show, is your drug superior to the standards of care, or at least comparable, or does it work on patient populations that the standard of care does not work on? From this, then we can go back to the Food and Drug Administration and ask for an NDA. This is going to be your new drug application. Your new drug application will allow you to enter the market to begin prescribing this as physicians, or to begin bringing this to your patients from the pharmacy counter. So, and then this can lead to a phase 4. Sometimes the FDA will ask you to run additional post-market safety monitoring, or run additional indications to see, does a drug in this indication also work in its separate indication? So this is going to be our phase 1 clinical trial. This is an older paper. This study was completed in 2013, so about a decade ago. However, I still think it's a really good case study showing this kind of boots on the ground hematology, oncology, the basic scientists working with the pharmacists, working with the clinicians in order to save some patients' lives, some patients that would otherwise be dead because of their refractory disease. So this led to a phase 2 clinical trial in 2014 in 30 patients. And this was just an astounding study published in the New England Journal, showing that 27 out of 30 patients, 90%, were in complete remission after the first month. So using this drug is that you had a 90% response rate where patients were in complete remission after infusion. And for oncology, particularly patients that are refractory to standards of care, this is astounding. This was the first study where this type of efficacy was ever seen. Pretty remarkable. So in 2017, this was scaled up even larger to a phase 3 clinical trial. In this clinical trial, we saw that the objective response rate was 82%, complete response rate was 54%, and over a year later, about half of the patients, 40%, had a complete response. Overall survival at 18 months was 52%. So again, just amazing data, again, changed people's lives. And because of this, this drug won FDA approval in 2017. So this was now you can prescribe it, Yescarta, by Kite Pharma. So it's a pretty fascinating story, and we're kind of learning how this happened right at the beginning. This drug, UCI, is rapidly expanding our capabilities as far as cGMP manufacturing for cell-based therapies. So if you're interested in being involved in this type of research, this is an amazing time to get involved at UCI, because right now this manufacturing facility, cGMP, means current good manufacturing protocols. These are FDA requirements as far as drug reproducibility and a number of different manufacturing protocols. And so these are now being developed at UCI, and this can be prescribed to patients. So it's, again, a pretty fascinating story. I hope you like this story as much as I do. So the way this works is that T cells, so CD3 is a general marker of T cells. This includes both your helper T cells that are very good at pumping out pro-inflammatory cytokines to help kill bad things like cancer, as well as CD8 positive T cells that have the direct cytolytic effectors. things like perforin and granzyme that help directly kill things like cancer. So it's pretty interesting. So these chimeric antigen receptor constructs, this DNA that contains that modified chimeric receptor, how this was developed is this was packaged into a viral particle. What is this virus? This virus is a virus called human immunodeficiency virus, so HIV. HIV attacks T cells. So in order to get their construct into T cells, what they did is the scientists took the HIV, deleted all the virulence genes, and kind of used it as a packaging tool to deliver a good gene, in this case, the chimeric antigen receptor gene, into the T cells. And then that would cause, these viral particles have receptors for the T cells. This allows the virus to then fuse. And instead of releasing HIV DNA into the cells or HIV nucleotides into the cells, this is going to release the chimeric antigen receptor nucleotides. And this will then get integrated into the gene, into the nucleus. And so then from there, you'll have, this processing and this machinery called transcription and translation, where you will now be expressing this modified chimeric antigen receptor. This will then be transferred back into the same patient. In this case, now these T cells are engineered to have specificity for the tumor antigens, the tumor markers, and will hopefully cause a durable remission in these patients. So the goal of this lecture is to kind of introduce you to this paper, make this paper a little more digestible prior to going to the quiz. So with the quiz is that I'm not going to give every answer to the quiz in this lecture, but hopefully I can kind of help guide you through some of these complex hematology oncology figures. So let's kind of walk through this, is that we have 5 different patients, 1, 2, 3, 4, 5. So all of these patients, their age is shown here. Their FISH, fluorescent in situ hybridization and cytogenetics, is shown here. So what this means is that if you look inside the bone marrow of these patients, is that some people, just looking at them using this technique called FISH, everything looks okay. But for some of these patients, particularly patient 4 and 5, is that these patients have weird things going on in their bone marrow. So that if you look inside the chromosomes in the bone marrow, you're seeing all these weird translocations. You're seeing chromosomal deletions. So essentially, there is something totally messed up with the DNA of these cells. And that's why the bone marrow is spewing out cancer. It's because there's something wrong in the nucleus. This is giving rise to cancer. And so with these patients, you could kill off the cancer, but killing off the cancer won't be good enough because the DNA is messed up. So in this case, patients with relapsed B-cell acute lymphoblastic leukemia have a dismal prognosis. The only hope for these patients is to get rid of the cancer to achieve a second remission with no minimal residual disease, so no disease detectable at all, and then get a stem cell transplant. So the hope for long-term survival is this stem cell transplant. So let's just kind of walk through the stem cell transplant. But before we get there, let's talk now about the rest of the table. So each of these patients got a different initial round of therapy. So these are chemotherapies. And so these are different protocols that are favored by certain institutions and certain clinicians over others. And you will have to know these, not for this class, but if you pursue hem-onc, you will have to understand these different protocols and the advantages and disadvantages. We're not going to focus here on that here, but essentially with these patients, they were given this initial round of chemotherapy, and for everybody it worked. They went into remission. They went into clinical remission. So that means no more detectable cancer. However, they then developed a relapse. The cancer came back. So that's what you see here, the duration of clinical remission 1. For some patients, it was about, a couple months. Other patients, not so great, just a little over a month. Some patients almost a year. But what you see is that, again, this cancer is aggressive and it came back. So what do the clinicians do next? Is they put them on salvage therapy. So we have first-line therapy, and then we eventually get to salvage therapy. Salvage therapy is essentially just throw the kitchen sink at the patients and hope it works. So with that, there is massive side effects associated with the salvage therapy, but in this case, it's tolerated because the only other option for these patients is hospice and, ultimately death. So what is the response to the salvage therapy? Some patients are MRD plus, some patients are MRD minus, some patients it didn't work at all and they can continue to have refractory disease. This is relapse disease, so detectable cancer, or 63% of the cells are cancer. And then what happens is that now the patients got on the experimental therapy, the T-cell therapy. All these patients developed good response. They went back into remission with no minimal residual disease. And then this was the outcome for each of the patients. So that outcome was the allogeneic hemopoietic stem cell transplantation, allo-SCT. So let's go into that a little more detail. So how does this work? So this patient has cancer. So what you want to do is first you want to get rid of the cancer, have no minimal residual disease. So after that, this patient has no immune system. For most people, that would be an issue, but for these patients, that's a good thing because their immune system was mutated and causing cancer. So here you want to totally wipe out the immune system, and then a donor would donate their bone marrow cells. So this is stem cells derived from the bone marrow where the stem cell populations reside. You'd get a stem cell infusion, and then what happens is that over time, those stem cells would grow and that would repopulate the patient with a new hemopoietic system. So they'd have new bone marrow cells, they'd have new red blood cells, new white blood cells, new platelets derived from that donor. And that's good, again, because they had mutations in their original bone marrow. So originally you want to get rid of the cancer, then you do an engraftment. There's a critical period for infections because these patients are immunosuppressed because they don't have an immune system or the immune system is just beginning to develop. There is risk for graft versus host disease. Graft versus host disease is not a good time and it is a major risk factor. However, the protocols are getting better for these stem cell transplants and the risk for graft versus host disease is less, even though it still does occur in about 5% of patients. And then eventually the immune system will recover. So how does that happen? Is that these stem cells that are transplanted have the ability to build out our entire immune system tree. So we can develop lymphoid cells, causing lymphoblasts and white blood cells, myeloid cells, giving rise to ultimately our innate immune cells. In particular, here's a neutrophil, platelets, red blood cells. So the allo-HSCT will rebuild the patient's blood and immune system. So making a BALL diagnosis, you're going to see different terms, and I want to walk through these terms in this slide. So refractory disease, clinical remission, medium or complete remission, minimal residual disease. What is all of this? So I built this little tree here where at the maximum level, where you have the most amount of cancer cells, this is bad. Ultimately, you want to have no cancer cells down here. So if you have the maximum amount of cancer cells, this could be at diagnosis or at relapse. This means it's detectable by any technique. So you have cheap techniques that you can run, or you have expensive techniques. So these are diagnostic techniques. So for these patients, typically you would run the easiest test. You could run something like FACS, fluorescent activated cell sorting. So this is your flow cytometry. So you could detect it by FACS. You could see the visible cells, the cancer cells. histology. So this could be, put a blood smear on a slide. Oh my gosh, there's so much cancer. And then ultimately a more detailed analysis, the deep sequencing PCR. You could pick this up. So if you have a lot of cancer, you are not eligible for a stem cell transplant, because I would take that patient, undergo this complex, expensive, traumatizing stem cell transplant procedure, but ultimately that patient would still have cancer, and so the cancer would just dominate and overwhelm the new immune system, and the patient would still have a very poor outcome. Let's say I give a therapy. This therapy might put the patient into complete remission, and so that's a good thing. However, these patients that are in complete remission might have some minimal residual disease. What is that minimal residual disease? So that means this would be undetectable by our less sensitive methods. So flow cytometry, histology, the blood smear, all of this, I wouldn't see any disease. However, when I do my more detailed molecular diagnostic, my deep sequencing PCR, polymerase chain reaction, at that point, I see this deep sequencing PCR, I still see some residual cancer. And that means that even though it looks good from a gross level, looking at my less sensitive diagnostic techniques, when I'm doing my more sensitive diagnostic techniques, there's still cancer. I haven't been able to totally wipe out the disease. So the goal of therapy is to get patients MRD minus. So that means it's undetectable by FACS, histology, or PCR. So in this case, now this patient is eligible to undergo the stem cell transplant. So that is ultimately the goal of this therapy. So a couple things to notice. Let's just go back a couple slides. So here, this patient, after salvage therapy, this patient is MRD plus. What that means is that this patient is ineligible for that stem cell transplant. After the drug, after the T cell therapy, now they are MRD minus. I've killed off those last few cancer cells. Now I can get the stem cell transplant. That's great. This patient here was MRD minus after the salvage therapy. So the salvage therapy works. There was no residual disease. This patient technically is eligible to get a stem cell transplant today. They didn't need the T cell therapy. They could have gotten the stem cell transplant right then after salvage therapy. However, in discussion with their doctors, in discussion with their clinician and clinical teams, this patient decided to go forward with the clinical trial, get this T cell therapy to make sure all the cancer was wiped out 100%. What do I mean all the cancer was wiped out 100%? Wouldn't MRD minus mean all the cancer is wiped out 100%? And the answer there is a little confusing. It's kind of yes and no. What do I mean by that? Is that in that biopsy, there was no detectable cancer. However, there might be something else in the body. For example, let's say I go in with a needle, pull out a bone marrow aspirate, there's no cancer detectable there. If I did a different biopsy at a different site, there's a small chance there could be some cancer left over. And so with that, essentially you can only analyze what is in the biopsy. You can't necessarily analyze the entire person. You can only make those diagnostic calls about what is in the biopsy. So here, this patient was MRD minus. They decided to go forward with the stem cell therapy or with the T cell therapy. They were then MRD minus, and then now they were eligible and decided to go forward with the stem cell transplant. What about patient 2? So I have 1 and then I jump to patient 3. What happened to patient 2? So patient 2 got salvage therapy and was MRD minus. That patient then decided, in consultation with their doctors, that patient then decided to undergo stem cell transplant and to not go forward with the T cell therapy. So that patient was excluded from this analysis because they just went forward with the stem cell transplant as soon as they were eligible and did not go forward with this clinical study. Look at patient 4. So patient 4 is interesting because patient 4, after the stem cell therapy, they were MRD minus. So they were eligible for stem cell transplant. However, there was still some residual cancer hiding somewhere in that body. And within 90 days, unfortunately, this patient relapsed. This patient was now ineligible for a stem cell transplant. And unfortunately, this patient since died because they were ineligible for that stem cell transplant, and therefore there was no hope for long-term survival. Oh, okay. So I guess I had that again. So anyways, table 1, patient characteristics and response summary. Which column represents the patient's disease state when they are enrolled in the clinical trial? It's going to be here. So the disease response to the salvage therapy is right here. And then they enter the clinical trial. Okay, so let's look at this cytokine release syndrome, this cytokine storm. So we know cytokine storm from coronavirus, but here we're seeing cytokine storm in response to therapy. In this case, this immuno-oncology therapy caused cytokine storm. What is this cytokine storm? Cytokine storm is a massive amount of cytokines. So here I have interferons, I have interleukins, I have IP-10, which is a pro-inflammatory chemokine. Essentially, what I have is I have massive amounts of inflammation. So this inflammation, I can see it. I can see it because of the cytokine levels in the plasma of these patients. In particular, patients 4 and 5 had a ton of cytokines, and that was associated with, again, poorer outcomes. What do I mean poorer outcomes? So here is that this is their temperature. These patients ultimately developed very high fevers. So when I'm doing a phase 1 clinical trial, the ultimate thing, the most important thing is I don't want my drug, my experimental therapy to kill the patient. You know, for this case, clinically, it's okay if the cancer kills the patient. Why is it okay? It's because these patients were refractory to salvage therapy. There was essentially no hope for these patients before they entered the trial. But these patients have some time left, time that they could spend with their families, time that they could spend, putting together their wishes and things like this. However, if you enroll them in a clinical trial and your experimental drug ends up killing the patients, that is not a good thing. That is, just, terrible, as you don't want to bring in a drug to kill the patients. However, we know that cytokine storm, having these incredibly high elevated fevers, will cause neurotoxicity, will cause, systemic organ failure. It's just terrible. So how do we shut down that fever? How do we shut down this fever? And essentially, we don't want the fever, we don't want the cells that we transfer to kill our patients. So what are we going to do? We're going to do this asterisk. What is this asterisk? This asterisk is the kill switch. So I can turn off the T cells. How do I turn off the T cells? Is I know the basic science that we have covered in the lectures to date. So how do I turn off all of this stuff? Is that these chimeric antigen receptors work by activating NF-kappa B. That NF-kappa B is going to drive the cytokine production. That cytokine production is causing the fever. So in this case, I can give an off switch. What is this off switch? The off switch is cytokine production. To drop that cytokine production, it's steroids. I can give corticosteroids, in this case dexamethasone, a potent anti-inflammatory steroid that's going to block NF-kappa B signaling, that's going to block cytokine production. And by blocking these pathways that I know exist in the T cells, I can drop the cytokine production, I can drop the fever. But there is a further downside, is that I can also blunt the pro-inflammatory and anti-cancer efficacy of this therapy. So that's ultimately what I'll see, and I'll go into that in a couple slides. So I want to talk more about these chimeric antigen receptors. So what are these signaling pathways? Okay, so T cells, we know T cells are the most important cell in the body. How do we know T cells are the most important inflammatory cell in the body? First off is through experiments like this, is I can give modified T cells and those can totally wipe out cancer and save people's lives. That's some pretty good indication that T cells are the most important inflammatory cell in the body. What else? Is I know this from immunodeficiencies. So for example, if I take a virus like a human immunodeficiency virus that kills off T cells, I give that, even though all the other immune cells are still functional, these patients will develop immunodeficiency, acquired immunodeficiency syndrome, AIDS. And so therefore, if I just kill off the T cells, the entire immune system has no idea what to do and therefore is susceptible. You can also see this with congenital immune deficiencies. So if I have mutations in T cell genes, is that this can give rise to severe combined immunodeficiency. So again, even though all the neutrophils and things like that are still there, without the T cells to tell them what to do, those cells are less effective at killing and protecting the body from pathogens. So T cells are so important, but I have checks and balances on T cells. What is the major extracellular check and balance on T cells? It's going to be MHC class 2. Typically, my T cells will only recognize antigen presented in the context of MHC class 2 in the case of CD4 cells. In the case of CD8 positive T cells, it would be antigen expressed in the context of MHC class 1. And essentially, I need an antigen presenting cell to talk to my T cell and make sure the T cell knows that it's okay to get turned on with an appropriate initial signal event, a co-stimulatory signal, as well as cytokines to provide information, this kind of three-step process to turn on these super inflammatory cells. So I don't want to turn these on unless the T cell is sure. However, this is a limitation to the T cells when you really want inflammation, like in the case of these patients. So what did Carl June at UPenn do in order to get rid of those checkpoints, to get rid of those breaks, to really unleash the full power of the T cells? The way Carl June did that is he liberated the T cells from the MHC checkpoint, from the MHC barrier. How did he do that? Is with these chimeric antigen receptors. So with the chimeric antigen receptors, these receptors here that you can see here, right now we're using a second generation. This is what this paper is using. This is not a T cell receptor. This is an antibody domain. So antibodies are free to bind anything. There's no MHC requirement for binding antibodies. Here he used an antibody domain, an antibody that's targeted against CD19, taken that antibody and put it into the T cell. And then what he did is he combined it using molecular biology techniques to combine it to CD28, to CD3, and essentially gave the normal T cell receptor and CD28 signaling power that's associated with normal T cell activation, but liberated it from MHC by putting it on this chimeric antigen receptor where this extracellular antibody domain can now bind directly to the cancer. And I don't need CD19 presented in the context of MHC class 2. In this case, I have a T cell that can now directly bind to the cancer cell and get activated through all these kinases that are regulated by CD3 signaling, by CD28 signaling, and this can activate my NFAT and NF-kappa B to drive cytokine production and cancer killing. So essentially, it's just a fascinating piece of molecular biology, somebody who really understood those signaling lectures that we had in this class, and they were able to use that information to really make a difference in so many patients' lives. So, examination of bone marrow biopsies by flow cytometry. So pre-treatment, patient 4 and 5, you can see they have tons of refractory disease. You can see there's a 45% of the cells here are CD10 positive, CD19 positive. Here in patient 5, 79% of the cells are CD19 positive, so tons of cancer in these biopsies. That's not great for the patients. Important thing to notice and kind of look at here is that these cancers are a little different, right? Is that these patients, they don't have identical cancer. This patient 4, their cancer is CD10 positive, CD19 positive. Patient 5, it's CD19 negative and CD10 positive. Or sorry, CD19 positive, CD10 negative. So the fluorescence is high for CD19, but not high for CD10. What does that mean? That means that these patients' cancers are not identical. These B cells became malignant at different stages in that hematopoiesis that we discussed. So these T cells, these B cells became malignant at different stages in development. But the thing is, for this therapy, we don't care. We don't care because it's a CD19 targeted therapy. As long as the cancer has CD19 on its surface, the therapy should work because it kills every single cell that is CD19. It'll hunt out, find CD19 positive cells, and then selectively kill those cells, regardless if they're CD10 positive or not. So now let's look at table 2. Table 2 is that deep sequencing PCR. So how do I know through deep sequencing? How can I use this as a diagnostic? So every immune cell is different. Every T cell, every B cell is different genetically. And this is fascinating because for most cells in your body, almost every cell in your body has the exact same DNA as every other cell because, this is just kind of what we're born with. One of the major exceptions is T cells and B cells. Why is this the case? Is because T cells, B cells have this fascinating VDJ recombination. The VDJ recombination causes unique sequences that cause the production of unique antibodies. So originally during development, when we're young, there's one non-combined IgH sequence. This is your immunoglobulin heavy chain. This is one of the pieces of the antibodies that's undergoing this recombination. So during development, there's going to be a variety of sequencing, a variety of changes to the DNA caused by this recombination, and you're going to generate many different IgH sequences. Ultimately, in development, some of these will be eliminated because of clonal deletion, because they're self-reactive, and then you're going to develop this mature pool of naive lymphocytes with many different IgH sequences. However, in the case of antigen encountering, you're going to clonally expand the ones that are producing the antibodies that you need to fight the bug. In the case of cancer, if one cell becomes cancerous, that cell is going to begin to divide over and over and over and over again. And therefore, all of these cancer cells will have the exact same IgH sequence. And because of that, if I'm picking up more and more and more and more IgH sequences, I know that these cells are all the same. These cells are all derived from the same malignant cell. These cells are cancer. So here, the total number of unique IgH arrangements that are detected, or the total number of IgH rearrangements that are detected by the deep sequencing, the PCR sequencing on the bone marrow biopsies. This is our sensitive technique that can help us determine minimal residual disease. So patient 1, this patient had some MRD. So of this, there were 57,000 B cells detected in that biopsy. Of those, 58 of those cells had the exact same sequence. Therefore, I know there's still a little tiny bit of cancer left. However, after my T-cell therapy, 55 days later, no cancer at all. Patient 3 only had 1,000 B cells detected, 1,000 IgH rearrangements detected, and none of those were the same as the rest. Therefore, this patient was MRD minus, with stem cell transplant eligible. They received the therapy. They had then no IgH rearrangements detected and no malignant IgH arrangements detected. Here with patient 4, you see that originally they had 2 million IgH arrangements detected, and almost all of those were exactly the same. So again, just a lot of cancer with this patient. After the therapy, less IgH arrangements, but again, there was definitely cancer detected, where a lot of these were the same. About half of these were all the same. Half of the total was about all the same. Patient 5, same thing, a lot of cancer to begin with. After the therapy, no more detectable cancer. So how do we monitor these? So when you're doing a clinical trial, the pharmacokinetics are very important. So understanding what's happening to the drug. So if I take the drug, put it into a patient, I need to know what's going to happen to that drug. In order to understand what's happening to that drug, I need an analytical method. For this case, in this paper, they use flow cytometry as their analytical method to look at the persistence of the cells. So these cells have weird receptors, chimeric antigen receptors that, again, are totally artificial. This is totally made in the lab. No cells in any patient, in any person on the planet have chimeric antigen receptors. This is something that's totally artificial and was designed to kill cancer for this study. And therefore, this stuff is totally unnatural. And therefore, I can monitor this by using antibodies directed against the chimeric antigen receptor. I can begin to pick up those cells that are circulating. Okay, well, thank you. I hope that's helpful. Good luck on the quiz. I wish you a lot of luck, and good luck in the class going forward.